You're welcome to class. We have microbial growth. Microbial growth is defined as the increase, right? Increase in the number of cells, right? And the cells we're looking at here is specifically bacterial cells. In some other studies, you can have fungi and some other related cells to study. Does that make sense? But we are looking at it in a population, right? What's the population? It's a geographical area where organisms of the same species live, right? They live there and they can actually interbreed. Does that make sense? All right. For growth to be aided, nutrient is required, right? And this nutrient can be in two forms. We can have broth and agar. So what's the broth? A broth is a liquid medium, right? What's an agar? An agar is a solid medium. Like for you to make a solid medium to become liquid is just for you to mash the medium, right? Just mash it, add some other solvents, right? That is suitable for that particular medium, right? For instance, in your study in the spontaneous generation of microorganisms, you must have read that some scientists made use of molten meat, right? So we have, if this is the meat, they boiled it, then they mashed it, right? And it became a liquid. Based on the availability of nutrients, microbial growth is divided into two systems. We have the closed system or the batch system, right? And we have the second system, which is the open system. In the batch system or the closed system, there is renewal of nutrients. Does that make sense? You try to get the bacterial cell in a vessel, right? Add the nutrients, whether an agar or a broth, right? On the addition of the nutrients to the microbial growth, you don't renew the nutrients then, right? You like close up the vessel or leave it open to observe the process. Does that make sense? Now, but for open system, we humans, for instance, we are operating the open system of nutrition, right? Now, once we eat, we pass out the waste product, then we eat again, and because there is renewal of nutrients. Does that make sense? So that's the difference between closed system and open system. So in a closed system, you observe about 5 stages rather. You have number one, your log phase, number two, your lag phase, or what you call the number one, your lag phase first, right? Then you observe your log phase or what you call your exponential phase, right? Right. Number three, you observe your stationary phase. Number four, you observe your third phase, you observe the third phase, right? Then you observe the phase of prolonged decline, the phase of decline, right? These are the 5 phases you observe in the closed system, right? If that is the case, let's quickly take the definition of closed system. The system in which is a system in which, right? It's an error. Is that comes to the A, right? Fine. Is that which does not require a renewal of nutrients, and in this phase you have the first phase, the lag phase. Now, once a microorganism or a bacterial cell is introduced into a medium, right? A nutrient medium now. Does that make sense? The first thing that will happen is that the microorganism would try to, or the microbial cell would try to adapt to the nutrient because that is not previous its previous home, and that is not the previous nutrient it's actually feeding on. Does that make sense? So the first thing is it will try as much as possible to adapt to the nutrient, right, to the new medium. Does that make sense? The second thing is that is to adapt to the environmental factors, right, like oxygen availability, temperature. The vessel inside this vessel might be a bit hot or might be warm, might be a bit cold, and depending on the temperature of the laboratory in which the experiment is carried out. Does that make sense? But it is unlike the bacterial cell being in the river, do you understand now, where it is actually collected from and the likes of it. Does that make sense? All right. The next thing the bacterial cell tries to do is to get some things done, right? It's just like someone who is traveling abroad. Once you get there, the first thing is you try to get used to their food, right? The next thing is the environment. The environment would have changed from hot to cold or cold to hot, right? Now, the next one is you try to know certain places you can get work before you try to increase in wealth, to try to increase in certain activities. Does that make sense? The same thing applicable to a bacterial cell, right? The next thing they do is they synthesize micro macromolecules, right, for multiplication. So the multiplication is observed in the next phase, and that is your log phase. Your log phase. So in the log phase, what happens? The log phase is also called the exponential phase, right? At that stage, it is actually called the exponential phase because the word exponential means index, right? So it can be 3 raised to power 4. 3 raised to power 4 is a big value, right? That's about 81, right? It's actually a big value when you take the exponential of a value. Does that make sense? Right. So there is a big increase there, and that's why it is called the exponential phase. So the first thing that is observed there is you observe a kind of rapid increase in the number of cells, right? Now, when the cells increases, what do you observe? There will be utilization, there will be rapid utilization of the nutrients because the more the population of the country, right, the more the more the food consumed by the people. Does that make sense? The same thing applicable to a bacterial cell in a plant system, right? Now, once that is done, what happens? Metabolism becomes faster, right? Because there will be synthesis of primary metabolites like nucleic acid, right? Like amino acid, and so on forth, right? Now, the third stage is your stationary phase when food is exhausted, right? So the nutrient is exhausted. Now, it is now between death and life because nutrients just got exhausted. Does that make sense? And if nutrient is exhausted, it means that there will be hunger, right? And some cells that are not fit for that condition are likely to die. That's the death rate. There will be death and there will also be reproduction. Why is death rate equals to reproduction rate here? Because some cells that are dying would be producing certain metabolites, right? They will be producing nutrients for other cells that are alive. Those cells that are still alive are called the viable cells, and those cells that are dying are called the dead cells. Does that make sense? Now, at that stage, the the nutrients they are releasing, right? The nutrient they are releasing is usually peptide and nucleic acid, right? For what? For the viable cells to be able to reproduce. Does that make sense? Right. Now here, the last thing observed is that spore formation, spore formation is usually dominating, right? Or sporulation is dominates, right? So the stationary phase in summary, three things observed. This is the stationary phase here, right? Three things observed in the stationary phase. Number one, there is death of organisms, right? Number two is that some cells leave, the viable cells. Number three is that spore formation dominates. Does that make sense? Now, those that are dying is just a summary. Those that are dying produces peptide and nucleic acid. Does that make sense? For what? For the viable cells. And once they do, these ones use these two things, two components here and here to reproduce. Does that make sense? And that's why death rate is equals to reproduction rate. Does that make sense? All right. Then another thing is spore formation. Formation of spore is just to protect themselves from dying. Does that make sense? From the unfavorable condition. And sorry. Now, in summary, the five steps that is observed in batch phase is actually abbreviated into D-cellar. D-cellar. Does that make sense? So I have D-cellar. The D is doubled here, right? And we have cellar, right? You pick from the back, right? The first phase is this. You have the first stage, LA, that's your lag phase. E is your exponential phase. S is your stationary phase, right? So we have picked number one, number two, right? Number three, right? The next one is your third phase, and the last is your phase of prolonged decline. So this is your phase of prolonged decline. That is all about it. So we have DCELA for the acronym for the stages that you observe in open system, right? Now, let's quickly observe the growth curve. The growth curve. You might be asked to draw this in the exam, right? And how would you draw it? You don't need to cram the curve, right? Let's draw it together. Let's draw it together. So this is what you have. So I have this two lines, right? Right here, you have the number of cells. Does that make sense? The number of cells. Here you have your time. The time can be in hours. It can be in days, right? In hours or in days. In hours or in days. Does that make sense? All right. So what happens in the graph? You have the first thing that happens to the cell is actually the lag phase. As at lag phase, nothing is increasing. Does that make sense? After that, there is an increase after it has synthesized the macromolecules for increased sake. Does that make sense? Is an increase. That the stage will be your exponential phase. Does that make sense? Exponential. Because the cells are actually multiplying at that stage. Does that make sense? Now, after the exponential phase, what happens at the exponential phase, or what you call the log phase, right? What happens at the exponential phase? Food is utilized fast. Does that make sense? After the food has been utilized, the food finishes, right? And once the food finishes, they get back to the stationary phase. Now, at the stationary phase, what happens at the stationary phase? Death is equals to reproduction, is equals to life. Does that make sense? By the production rates. Do you understand now? By the viable cells. This ones produces some nutrients like peptides and nucleic acid for these ones to live. Does that make sense? If that was the case, this stage here, you observe a straight line here. Why? Because it is neither increasing nor decreasing, because at that stage, they are just dying. But while something is trying to bend it down, by death, right, one is also raising it up. And in the end, they attain an equilibrium position. Does that make sense? And that's why it is straight like that, right? So we have this as our stationary phase. So I can experience stationary phase here. Does that make sense? So this is your stationary phase. So after that, what happens? The condition becomes unbearable. Do you understand now? What will happen? More death is recorded here. Does that make sense? And that is actually, we've not talked about the third phase. That's actually your third phase, right? This is the third phase, right? So there's recordings of more cells that will be dying. Does that make sense? Now, immediately after that stage is a stage of prolonged decline where the cells does not only die, right, and some try to survive. Does that make sense? Most of them dies, right, and that's the phase of prolonged decline. Does that make sense? All right. So what happens at this stage? At the stages we're talking about, number one, there is depletion of nutrients, right? Rapid depletion of nutrients, right? Then the death rate increases, right, and exceed the reproduction rate. Then there is, as the more they are dying, there is release of toxic materials into the cell, into the vessel, and sporulation increases because some will thrive to live at all costs. Does that make sense? But at the last stage, which is the phase of prolonged decline, what happens? The condition becomes more unfavorable, and there's lack of nutrients, there's lack of food, there's lack of strength, and the likes, and every other unfavorable condition that you might think of as a human dreams now exist in that vessel. And then there is a high rate of what? Of decline. All right. That is that for growth phase 1.